This week is going to be your first genetics lecture. I have to tell you, I love genetics, so, forgive me for gushing. But the first thing we're going to talk about is Mendelian genetics, and then we're going to talk a little bit more about inheritance and then DNA itself. We did talk about meiosis, which is when cells divide into haploid cells, and for humans at least, it's only our reproductive cells that do that, so that does kind of run directly into this because that cell division during meiosis is what's going to allow the gametes to come together and create a new organism that will have inherited traits from its parents. So, what genetic principles account for the passing of traits from parents to offspring? If you haven't noticed, you may share traits with your biological parents or your biological siblings. So, why are those traits passed? How do parents pass those traits on to their offspring? The blending hypothesis is the idea that genetic material, so DNA, from the two parents blends together, the way that blue and yellow paint will blend together to make green. So each one of those gametes that underwent meiosis is going to come together, create a new 46 chromosome human zygote, and that will then create new traits because it's going to blend the traits of the parents. The particulate hypothesis is the idea that parents pass on discrete heritable units, genes, and that is more accurate to what is going on. Mendel documented a particular mechanism through his experiments with garden peas. So Gregor Mendel was the guy whose picture was at the beginning of this lecture slide. He lived, a few hundred years ago. He was a monk, but his job as a monk gave him enough time to do some really great genetics experiments using pea plants, so like green snap peas, those type of plants. So you can hear a lot about plants for a second here. Mendel discovered the basic principles of heredity by breeding these garden peas in carefully planned experiments. He used a quantitative approach. So Mendel probably chose to work with peas because there are many varieties with distinct heritable features, so they do have different flower colors, and character variants. And these are their traits. He would control the mating between the plants. So plants can reproduce by using pollinators. They can also reproduce using wind. Or if you're an enterprising gardener, you can help your plants by cross-pollinating individuals yourself. He chose to track only characteristics that occurred in two distinct alternatives. So it was a yes or no thing, none of this blended stuff. He also used varieties that were true breeding, so plants that produce offspring of the same variety when they self-pollinate. Plants are able to self-pollinate, which would be a form of sexual reproduction, though it's their own gametes that are coming together. And when he chose plants that when they would do that, they would produce the same variety, so they would always have the same ones from doing that. How did this work? So in a typical experiment, Mendel mated two contrasting true breeding varieties, a process called hybridization. So he would take one plant as the parental generation, which we call the pea generation, and he would cross-pollinate one true breeding plant and another, and then he would look at their first generation of offspring. The true breeding parents are the pea generation, and the hybrid offspring are the F1 generation, and we still use this nomenclature. So this would be the P generation and then the F1 generation. When the F1 individuals self-pollinate or cross-pollinate with other F1 individuals, so we would cross-pollinate two of these plants, the F2 generation is produced. So let's look at the law of segregation. When Mendel crossed contrasting true breeding white and purple flower pea plants, all the F1 hybrids were purple. So from this white plant and this purple plant, he only would get purple plants. When he crossed the F1 hybrids, many of the F2, so this next generation, had purple flowers, but then these F2 plants would also have these white flowered plants that would reemerge from the parental generation. He discovered a ratio about 3:1 purple to white flowers in the F2 generation. So while the F1 generation was all purple plants, the F2 generation would see the reemergence of the white flowers. That's what you see here. There's not just one plant being produced in the F1, they just all look the same, so only one is pictured. And this ratio of 3:1 was consistent. He reasoned that the F1 plants, the heritable factor for white flowers, was hidden or masked in the presence of purple flowers. The purple flower factor. We have a phrase for this that gets really used commonly, sort of colloquially, and that's it skips a generation, right? So, oh yeah, we have red hair in our family, but it skips a generation. And so your grandparent might have it, but then your parents wouldn't, but then you would. So that's kind of what he's referring to. He called the purple flower color a dominant trait, which is a term we still use, and the white color flower a recessive trait, again, a term we still use. The factor for white flowers was not diluted or destroyed because it reappeared in the F2 generation. It wasn't as if that F1 generation was expunging that trait, it was just masking it. He observed the same pattern of inheritance in 6 other pea plant characteristics, each represented by 2 traits. So again, these are all yes or no characteristics. We will talk about characteristics that fall on more spectrums later on. But what Mendel called a heritable factor is now called a gene. The really cool thing about these experiments that Mendel did is that he did this not knowing about genetics. In fact, he's a contemporary, so he lived at the same time as Darwin. So Darwin is just starting to formulate the theory of evolution. He's starting to write the origin of species by means of natural selection. These are going on at the same time, and they actually never spoke. If they had, it might have advanced the whole area of genetics really quickly, but they never spoke. But Mendel was doing these experiments around the same time, and he did all of this without knowing that there were what genetics were or what DNA was. Bearing in mind that DNA would not really come into the scientific consciousness until the 50s. So he's way ahead of his time. So he noticed these ratios were always the same. And here's some more of those yes or no traits. He would always see the same ratio. And these are exact ratios here, but you're not going to get 0.15 of a flower. He would see something very close to this 3-to-1 in all of these traits. So if he had the seed color, he would see that there were a 3-to-1 ratio of yellow to green seed colors, or if they were round or wrinkled, inflated or constricted. So the model that he ended up coming up with was that he observed a model to explain the 3-to-1 inheritance pattern. 4 related concepts make up this model. The first is alternate versions of genes account for variations in inherited characteristics. So there are different genes that are creating different traits, different phenotypes is what that's called. For example, the gene for flower color in pea plants exists in 2 versions, one for purple flowers and the other for white flowers. This alternative version of a gene is called an allele. So that allele is still the gene for flower color, but there are 2 different types of that gene, which we call alleles. Each gene resides at a specific location on a specific chromosome, a specific locus. So the location on the chromosome is called the locus. For instance, let's say this is the chromosome for the flower. That gene for flower color is always on the same spot, but you might have 2 different variations of that. And you can see here, at one there will be an A and the other a C at the same location. So second, for each character, an organism inherits 2 alleles, one from each parent. Mendel made this deduction without knowing about the existence of chromosomes. So we figured this out without knowing chromosomes existed. 2 alleles at a particular locus can be identical, as in the true breeding plants from the pea generation. So if they're identical in a white flower, then all the offspring are going to be white. Alternately, the 2 alleles at a locus can differ, as in those F1 hybrids. The third, if the 2 alleles at a locus differ, then the dominant allele, is going to determine the organism's appearance, and the other, the recessive allele, is not going to have a noticeable effect. That is not to say it is not present or that it is destroyed. It's there, it's just not really doing anything that we can see. In the flower color example, F1 plants had purple flowers because the allele for that trait is dominant. So that F1, who had a true breeding purple plant and a true breeding white plant as parents, they would have had a dominant allele, which is usually denoted with a capital letter, and a recessive allele. So they would have looked like that. Just really quickly, so if you've ever seen cats that have 6 toes, kind of cool. But Hemingway famously had cats with 6 toes, and my stepmom's cousin actually has some of the descendants of those cats, which is super cool. He's a vet. I think somebody gave him to him. But and Hemingway had a house in the Bahamas, and you can still go there today, and there's a colony of 6-toed cats. It's kind of weird. Dominant alleles are not necessarily more common in a population than recessive alleles. And going back to the polydactyly here, one baby out of every 400 in the United States is born with an extra finger or toes, a dominant trait called polydactyly. So for these cats in Hemingway's house, there's more of them with 6 fingers or toes. But for humans, that's not the case, though it is a dominant allele. So someone can have a dominant and a recessive allele for polydactyly, which is having an extra finger or toe, and still, and they would have that, but that does not necessarily mean that that's the common trait. So the next is the law of segregation. So we kind of discussed this last time, but this is a little bit more detailed in it. The fourth, the law of segregation, 2 alleles for a heritable characteristic are going to separate or segregate during gamete formation and end up in different gametes. So going off those cats, let's say one had and I'm going to underline the capital letter. They were going to have a dominant allele for polydactyly and a recessive allele. These 2 alleles, they're going to have 2 different gametes, right? One is going to get the dominant one, and the other is going to get the recessive. So they're going to separate during gamete formation. Any egg or sperm gets only one of these 2 different alleles that are present in the organism. So saying that polydactyly in cats, even in humans, is rare, this one with the recessive allele is probably more likely in a breeding population to encounter an individual who does not carry this trait, and then they would have offspring that weren't going to have polydactyly. Now, if they are the gamete that have the dominant trait, and they encounter an individual who has the recessive, they could still have offspring that have polydactyly. The segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. Remember that random assortment thing? It's that. In order to determine what the outcome for different breedings would be, you can use something called a Punnett square and then just use ratios. So Mendel's segregation model accounts for a 3-to-1 ratio in that F2 generation. The possible combinations of sperm and egg can be shown using a Punnett square, that's this thing here, to predict the results of a genetic cross between individuals of known genetic makeup. So if you are a individual with a dominant and a recessive allele, actually let's use a different trait though, right? So we're going to go back to the purple flowers. I'm going to use F as that. So let's say you're a purple flower. You could have a dominant and a recessive trait, or you could have 2 dominant traits. So we don't know the genetic makeup there. So we really can't do this, but you could determine the parental genetic makeup, the genotype is what that's called, from looking at the offspring, however. And again, a capital letter is going to represent the dominant one, and a lowercase letter is going to represent the recessive allele. So for these purple flowers, you would have a big P and a little p, or the F's as I used here. I can erase all of this, clean that up a little bit. And what you see is, if you have 2 heterozygous, so they have a dominant and a recessive trait, you could have individuals that were homozygous dominant, heterozygous, which means they have 2 different alleles, or homozygous recessive. Let's pretend this is looking at the purple flowers. This individual here, this one here, and this one here would all have purple flowers still. So there's 3 of them. And then this final one here would have a white flower. So you'd be back to that 3-to-1 ratio. So you're going to have, with these 2, if you did a Punnett square, you'd have the purple flower here with their capital P's, and then the recessive phenotype down here with the white flower, and you'd have one individual, another individual, another individual, and another individual. All of them have purple flowers. But then down here, you see that because you have individuals running around that are heterozygous, the possibility is then that they could produce homozygous recessive offspring. Those would be capitals there. So here's that vocabulary. An organism with two identical alleles is homozygous, and you can be homozygous dominant or homozygous recessive. For an individual that has two different alleles, you are heterozygous. So unlike homozygous, heterozygous is not true breeding because, again, using the flowers, let's say you have a heterozygous individual that has purple flowers and you crossbreed them, you'd end up with 3 possible genotypes. But these two would be purple and this one would be white, so this individual would not be true breeding. But what's the difference between genotype and phenotype? Okay, so because of the effects of a dominant and recessive alleles, an organism's traits do not always reveal its genetic composition, because everyone's going to have one allele from mom and one from dad. Therefore, we distinguish between an organism's phenotype or physical appearance. So phenotype is referring to the actual physical trait that we can see, or in the case of disease, that manifests. It's the trait that manifests. Or so the physical trait, the physical appearance, and its genotype, which is its genetic makeup. For those purple flowers, the genotype here is heterozygous, but the phenotype is just a purple flower, and it's the dominant phenotype. I can make you all roll your tongues by doing this. So the dominant phenotype for rolling your tongue, you can do that. Non-rollers, that's recessive. But just because both your parents can roll their tongues doesn't mean that you'll be able to, because that would indicate that one or rather both of your parents were heterozygous. So if both your parents can roll their tongues, they must be carrying that dominant allele. But if their offspring cannot, they must also be carrying the recessive allele in order to produce offspring that can't roll their tongues. Again, with the purple flower, PP and P little p, so homozygous dominant and heterozygous, are going to have the same phenotype. They're just going to be purple flowers. You see that here. The phenotypes are 3 to 1. The genotypes are going to be 1 to 2 to 1. We can make this box a little bigger. That's the ratio of genotype. The phenotypic ratio is 3 to 1. So how can you tell the genotype of an individual with a dominant phenotype? We kind of discussed that just recently, but we're going to get more into it here. So an individual, such as an individual, could be homozygous dominant or heterozygous. If I see a purple flower, I don't know what their genotype is. It could be either one of these. The answer is to carry out something called a test cross. So a test cross, you're going to see the breeding of a mystery individual, which we're just going to call this purple flower here, and a homozygous recessive individual. Because if I have a white flower, I know they must have 2 lowercase p alleles. They must have 2 recessive alleles or be homozygous recessive. And you can't read that with that drawing there, so I'm going to erase that real quick. If any of the offspring display the recessive phenotype, then the mystery parent has to be heterozygous. Just to show you why that is, so let's get rid of all this. Mendel identified his second law of inheritance by following two characteristics at the same time. So now instead of just looking at flower color, you're going to look at flower color and seed shape, for instance. Crossing two true breeding parents, so that would be two plants that, let's say we're going to look at seed shape and there's going to be rounds and wrinkles, right? So the true breeding parent would have to produce offspring that when they were self-pollinated that only produced those seed shapes, just like the flower color. He would make these dihybrid crosses, and in the F1 generation, there would be individual or heterozygotes for both of these characters. He can determine then if two characteristics were transmitted to offspring as a package or independently. Looking at that, now you have quite a bit more phenotypes possible, right? Because you have not only the 3-to-1 ratio, but if you have two, what he would notice is this ratio of 9-to-3 to 3-to-1. So 9 of these individuals look the same. And then here's another 3 that look the same. 1, 2, 3, 4, 5, 6, 7, 8, 9. There you go. And then 3 that look the same. And then one individual that would be carrying 2 recessive genotypes, homozygous recessive genotypes. The results of Mendel's dihybrid experiments are the basis for the law of independent assortment. And it states that each pair of alleles segregates independently at any other pair during gamete formation. So just because you have a yellow seed that is round doesn't mean that all the yellow seeds that are ever produced have to be round. Some of them can be wrinkled. This applies to genes on chromosomes that are not homologous or those far apart on the same chromosome. So non-homologous genes, genes on different chromosomes, those chromosomes are independently assorting anyways, so it doesn't matter. What we know now is that if two genes their loci are very close together, they are likely to be transmitted to one another, especially if, and here's my sister chromatids, they're near the center, because it's just physically harder for that to happen. But again, we're going to have this phenotypic ratio of 9-to-3 to 3-to-1. So I have 9 round yellow seeds, yellow and round being the dominant traits. I have 3 shriveled yellow seeds, and they can either be heterozygous dominant or heterozygous or homozygous dominant for that trait. I have 3 green round seeds, and then 1 is just going to be wrinkled and green. It's going to carry both the recessive traits. So Mendel's law of segregation and independent assortment is going to reflect the rules of probability. The outcome of one coin toss has no impact on the outcome of the next toss. If I flip a coin and it lands on heads, and since I have a 1 out of 2 chances of getting heads or tails, just because I got heads the first time doesn't mean I'm going to have to get tails the second. I actually had a friend when I lived in LA. When you live in LA, it's like super easy to drive to Vegas. And I don't gamble because I don't want to hand someone my money and not get anything in return. So it's not like a moral judgment. I don't really care. I just don't like to just give someone my money. But my friend likes to gamble. So when we'd go to Vegas, I'd kind of hang out with her and get free drinks and stuff. And she would always think, like at the roulette table, that, oh, it's been hitting red a bunch, so it's going to hit black next. It doesn't matter. Every time they spin that wheel, it resets. The probability resets. In the same way, the 2 alleles of one gene segregate into gametes independently of another gene's alleles. Just because the flower is purple doesn't mean its seeds have to be wrinkly. Those are 2 different things. And for this, we're going to use some math. So the multiplication rule states that the probability of 2 or more independent events will occur together is the product of their individual probabilities. This can be applied to F1 monohybrid crosses. So segregation in a heterozygous plant is like the flip of a coin. Each gamete is going to have half the chance of carrying the dominant allele and half the chance of carrying the recessive. So let's say they have 2 heterozygous parents. Everybody has a 50% chance of getting either one of these alleles. So for that genotype, because these are really the same, you would have half the chance of getting that genotype and then 1/4 of a chance of getting either of these. And every offspring, individual offspring, has that chance. The addition rule states that the probability of any one of two or more mutually exclusive, so one can happen or the other, events will occur is calculated by adding together their individual probabilities. It can be used to figure out the probability that F2 plant from a monohybrid cross will be heterozygous rather than homozygous. One way to think about these is or and. Okay? And is multiply, or is add. We'll talk more about this during the Zoom call. Solving complex genetics problems, you can use probability to do this. You can apply these rules to the probability to predict the outcomes of two crosses. A dihybrid or other multi-character cross is equivalent to two or more independent monohybrid crosses occurring simultaneously. So to calculate the chances of various genotypes, each character is considered separately, and then the individual probabilities are multiplied. If it's or, you would add. For instance, if you have a 1 in 4 chance of having a dominant, homozygous dominant genotype for seed shape, and a 1 in 4 chance of having a homozygous dominant genotype for flower color, you just multiply those together. The chances that one offspring is going to be homozygous dominant for both traits is then 1 in 16. We can do these on and on and on and on. Now, if you want to do more than one, you can do this ad nauseum for as many as you'd like. So not all heritable characteristics, though, are determined as simply as Mendel's traits. Not all of them are going to be that simple. However, that basic principle of segregation and independent assortment universally apply to more complex traits. It's still that you're getting one allele from one parent and one from the other. Inheritance of characteristics by a single gene may deviate from simple Mendelian patterns in the following situation. When the alleles are not completely dominant or recessive. When the gene has more than 2 alleles. So some genes, like your hair color, are going to have, I'm trying to think of letters we haven't chosen yet, more than one allele associated with them. And when single genes produce multiple phenotypes. So alleles are simply variation in the gene's nuclear sequence. That switch of a C to a T is going to determine if you're going to have a purple flower, that's my purple flower, or if you're going to have a white flower. When a dominant allele coexists with a recessive allele in a heterozygote, they do not actually interact at all. So it's not like these two are going to create something different when you have a Mendelian trait like this, because for this trait, it's just kind of like he doesn't exist, even though gametes can get them and they can be passed on. For any character, dominant or recessive relationships of alleles depends on the level at which we examine the phenotype. There is something called degrees of dominance, and this would be that instance in which you can have homozygous individuals producing more than one phenotype, or really individuals that are going to have the dominant phenotype. They're heterozygous individual and a homozygous individual, dominant individual, are going to have 2 different phenotypes in this case. Complete dominance occurs when phenotypes of the heterozygote and the dominant homozygous dominant one are identical. In the case of those purple flowers, that would be this. Incomplete dominance, which is here, the phenotype of F1 hybrids is somewhere between phenotypes of 2 parental variations. So this would be if you had like a red flower and you had a blue flower, and that ended up producing purple flowers. It's a blend. Codominance, the two dominant alleles affect the phenotype in separate but distinguishable ways, and that's here. There is red there. There is white there. But instead of being pink, it's just spotted. And here we can see that. So this would be that incomplete dominance, where these two individuals are creating a separate phenotype that's a blend between the two. And in this case, you're going to have offspring that are red flowers, pink flowers, and white flowers. So now, instead of a 3-to-1 ratio for phenotype, you're going to end up with a 1-to-2-to-1 ratio, something much more similar to the genotype ratio. A human example of this with a disorder is Tay-Sachs. It's fatal. It's a dysfunctional enzyme that causes the accumulation of lipids in the brain. It kind of has global effects. On an organismal level, that allele is recessive. But at a biochemical level, the phenotype, i.e. the enzyme activity, is incompletely dominant. So there's a bit of a blend. And on the molecular level, those are codominant alleles. Another more common example is with blood type. So humans have either A or B blood markers, and these are just are markers on your red blood cells. So your body, and this is and/or, but your body doesn't attack your red blood cells. You either have these markers on there, and your white blood cells see these markers, and they go, okay, you are part of the body. We're not going to attack you. For most of these genes in a population, there's more than two allelic forms. For example, the four phenotypes. Now, the four phenotypes for blood type, for the ABO blood type, determine the 3 alleles of the gene. So what are the blood type phenotypes? You can have type A, and this is ignoring positive or negative. You can have type B, you can have type B, AB, or you can have type O. And type O blood types is usually indicated by an italicized I. The enzyme adds specific carbohydrates to the surface of the blood, so it's going to add these markers. Your body will then have antibodies in the plasma that are the contrary ones to the markers you have on your blood. So if you have type A blood, you have antibodies for type B blood. If you are type B, you have antibodies for type A. If you are AB, you don't have any antibodies. If you are type O, you have no markers, and so you have antibodies present for A and B. That is why people with type AB blood can receive blood from anybody, and then type O can give blood to everybody because they don't have any antibodies on them. Type O can only get blood from type O, though, because if they get blood from these other blood types, they're going to have blood clotting. So that's how this looks. Type AB has markers for both, and so they can get blood from both A and B, where O can only get blood from O, but they can give their blood to everybody. It's a little bit of an unfair system. There's another, oh, that's not a good highlighter color, is it? There's another type of inheritance called pleiotropy, where you have one gene that has multiple effects. For example, pleiotropic alleles are reasonable for multiple symptoms for certain hereditary diseases. Sickle cell is a great example of that. You literally have one gene that has one nucleotide that's wrong, and you have a global effect. So you have a single gene producing multiple effects. So looking at Mendelian genetics for 2 or more genes, you can have polygenic, which means you have many genes affecting one trait. Height is an example of that. Your height is your height, right? How tall you are is how tall you are. But there are multiple alleles or multiple genes that go into determining how tall you'll be. We call them markers. Alternatively, multiple genes can independently affect a single trait. So I think there's like 25 identified markers for height. You can get any combination of those. Because of that, when genes are polygenic, we often see this type of distribution where you have a bell curve, but you also have a contingency, right? Like you have a, you don't have yes or no, you have sort of variations in between. It's not like your height is a yes or no thing. You don't have a height or don't have a height. You can have, you can be 5 foot, you can be 5'1", you can be 5'2", and anything in between. So there's more of a spectrum that it will fall on. Epistasis, a gene at one locus alters the phenotypic expression of a gene at a second. So for here, you have a gene that is not next to or near that, or is not part of that same gene, but it's going to affect it nonetheless. In Labradors and many other mammals, their coat color, which we call your hair color, depends on 2 or more genes. So one gene determines the pigment, and we're going to call that B. And then the other is going to determine the pigment will be deposited in the hair. So with that, you end up having, so you have 4, 5, 6, 7, 8, 9, the same type of ratio as you would with those dihybrid crosses, but you also have 3 distinct phenotypes. You can have a black lab, you can have a brown lab, or you can have a blonde lab. You have 3 distinct phenotypes, but it's because one gene is affecting another. Polygenic inheritance is very common. Its quantitative characteristics are those that vary in a population. If you've ever tried to buy foundation for makeup, skin color falls on a wide spectrum range, and they never make foundation for the actual color of skin that you have, and it always never matches. But there are just so many traits involved in it that you can have a wide variation. And that also means that 2 full siblings can have variant skin pigments. My sister and I have that. I'm slightly darker than my sister is. This is not, of course, to mean that everything about the way an organism is going to be determined by its genetics. But that is not to say that genetics doesn't play a part in it, right? So here's the confusing part, which we dub nature versus nurture, and it sounds super simple, but it's not. So there is an environmental impact on your phenotype. You can have, and this is also actually what got 23andMe in trouble and why they had to change their marketing, but you can have the genes that make you predisposed to something, but if the environmental factors aren't there, it might not manifest. Good example, certain types of cancer. If you don't smoke, but you have a predilection to get lung cancer and you're not exposed to any carcinogens, it's very unlikely you develop it. Not to say you couldn't, but it's very unlikely. The norm of reaction is that you have this phenotypic range of genotype influenced by the environment. That's normal. That you're going to have certain genes that might predispose you to something, but if the environmental factors aren't there, then that phenotype doesn't manifest. Diabetes is like that. They used to be 100% convinced hypertension was like that, but I think now it's actually leaning towards that might be more genetic. But just bear in mind that there is this environmental factor that has to be present as well for some phenotypes, not all. Obviously, like your hair color is your hair color, and that's the hair color you have. There's really a multifactorial determination for a lot of our phenotype, including our behavior. The phenotypic range is generally broadest for polygenic, so things where you have multiple genes interacting for one trait. This is a little filthy. The genetic and environmental factors collectively are going to influence the phenotype. Environmental factors can influence disease, but genetics may give you a predilection for that disease. Therefore, that's why you want to take a family history when you diagnose somebody. Does that mean that just because someone in their family had it, that's what they have? No, because their family could have been misdiagnosed. Also, again, this is what 23andMe is doing. It's not telling you have something. It's telling you have a trait for something. Really, this is probably also where a lot of mental health diagnoses lie, is that there is a multifactorial outcome for that, or multifactorial causative of that. And so the Mendelian view of heredity and variation is that an organism's phenotype includes its physical appearance, its internal anatomy, its physiology, and its behavior. All of these are your phenotype. Its phenotype reflects an overall genotype and unique environmental history. For certain organisms, now that, again, that's not to say that things aren't learned either. You have all sorts of learned traits. I'm teaching you stuff now, so you knowing this will be learned. But you have other learned traits too. So, I mean, why you might sound like your biological parents could be a determinant of like your facial structure is similar, as well as the fact that the same people, you know, that they taught you to talk, or that your sibling, your full biological siblings and you might sound similar, would be that the same people taught you to talk. But again, that would be right, like the biological phenotype of your facial structure and your vocal cords and your chest anatomy that would determine the sound of your voice, but also the learned environmental part of somebody taught you to talk. So an organism's phenotype is going to reflect an overall genotype and environmental history. This is also why you can't create a clone that is your copy. So if I have a person and I clone them, number one, I end up with just a baby, because a clone would be an infant at the time of their cloning. If I have an adult human, I'm going to end up with an infant clone that is genetically identical to them, but because the same factors won't occur to this clone, they're not going to be the exact same person. Many human traits do follow Mendelian patterns, however. Humans just aren't very good test subjects for genetic research. Number one, our generational time is really long. It's, you know, approximately average 20 to 25 years for a person to reach maturity. Parents produce relatively few offspring. Humans just don't have enough offspring to really check those ratios. And breeding experiments are just really unacceptable ethically with humans, so we don't do them. However, basic Mendelian genetics endures as a foundation for human genetics. In order to do genetic experiments, we often use a model organism, and that model organism is yeast or mice or Drosophila, which are these guys. They're fruit flies. They have a very short generational time. They have a lot of babies, and we can see them. Genomics is allowing us to now perform large-scale population studies for humans, and what genomics is looking at really big data sets and using computational power to calculate out which genes are involved with which traits. You can also do something called a pedigree analysis, and a pedigree is basically a family tree that describes the interrelationships of parents and children across generations and looks at a certain trait. So we look at these inheritance patterns for particular traits and can be traced and described using these pedigrees. It can also be used to make predictions about future offspring. If you become a genetic counselor, you use these a lot. We can then use multiplication and the addition rules to predict the probability of specific phenotypes. There are some, I'm going to change the color of pen I'm using so that it doesn't get confusing. How about that? We're going to use purple. So, or green, because it's very different. There are some basic rules for pedigrees. A square usually indicates a male, a circle indicates a female. For this one, they've been colored red if they're affected and blue if they're unaffected. Sometimes it's just that they are colored in if they're affected, and then being empty would be not affected. So a female, like I said, females are usually circles, males are squares, affected would be colored in, deceased, they have a line through them. Twins, you'd have the same setup of having the parents reproducing, but you do that to indicate they're twins. Adopted, because we wouldn't know their genetic history, and then a miscarriage. And this will inform us if a trait might not be something that is viable. So how do we determine the mode of inheritance? We're going to spend the rest of the lecture looking at this. If you want to take a break, do it now. So there are a couple ways to determine if something is autosomal recessive, if it's X-linked, which would mean it's linked to sex chromosomes. So looking at autosomal recessive. Recessive, again, you're going to have two recessive alleles, right? So if Oh, and this little dot in the center is that they are carriers, that little dot right here. An affected individual can have parents that are not affected. It also, in autosomal recessive disorders, does not appear in every generation. Here's that it skips a generation thing, right? That does not automatically mean it just skips. It just doesn't appear in every generation. And while this is a little hard to determine because there aren't a lot of individuals that are affected, we can say that males and females are equally affected. So you have an affected individual does not have affected parents. The trait does not appear in every generation. And from what we can tell, if we can tell, males and females are equally affected. However, that does not necessarily always happen with these because sometimes you only have one affected individual. An X-linked recessive disorder would be a disorder where you have a trait that's carried on that sex chromosome. So since females are XX and males are XY, males are known as hemizygous, meaning they only have one X chromosome. If something is linked on the X chromosome, it's more likely to affect males, because if their mother is a carrier, they're not going to get another X chromosome that can mask that disorder. So what we see with that is that males are affected in far higher numbers than females, but females are carriers. So in an X-linked disorder, you also will have, and I'm going to clean this up, fathers that do not, I said that backwards. You will have fathers that aren't affected with affected sons, and if you have an affected father, he will not have affected sons. He could have affected daughters, but most likely they're going to be carriers. Because if the father is affected, and we're going to say affected is going to be red. So here's the dad, and the Y chromosome is just the Y chromosome. Doesn't really factor into this at all. So I'm just going to make it a different color. And you have the mom, and she's a carrier, but she has a non-affected X chromosome. She could have a gamete that is carrying this, right? So her sons could only have that. It is possible that they, I'll do that so you know that's from the dad. It is possible that they get their mom's non-affected chromosome, and that's the case we actually see here, where you have one son affected and one son not affected. But if you have girls, so you have X and mom's other one that's not affected. If you have girls and their dad is affected, they have to get an X chromosome from both mom and dad. So they could get the affected phenotype, or they could get the affected allele, but then have the non-affected allele from mom. So really, these would be some of the options. The other option then would be to have a son who's not carrying it and is not affected. Okay, so how do you determine the mode of inheritance? For something that is autosomal dominant, this one, what you'll see is affected individuals must have affected parents. If you have a dominant trait, that's not a good color to use for that. Let's not use X, because that would be something else. If you have a dominant trait that one of you is carrying, or has, because it's the phenotype, because it's dominant, and even if the other person is not carrying it, the Punnett square you can still produce heterozygous offspring with the dominant trait, and thus they would have that phenotype. Also, autosomal dominant traits will appear in every generation. For autosomal recessive traits, what you'll see is, I'm going to get a good color that will actually show up here. Affected individuals may not have affected parents, and that would be because they are carrying the trait. So, I have two heterozygous individuals that reproduce. They still have a chance of producing a homozygous recessive individual. Another thing is, males and females will be equally affected. And it might not appear in every generation. For an X-linked dominant trait, so this is going to be something that's on the X chromosome, so you'll have chromosomal females will have XX and chromosomal males will have XY. What you'll see is that daughters are more affected because they're getting both X chromosomes, so there's a higher chance that they'll get it. And there is math to this. So for a female, she has a 100% chance of getting an X chromosome from her father. For an XY male, they have a 0% chance of getting an X chromosome from their father. So if their father is affected by this X-linked dominant trait, they don't have any chance in the world of getting it from their dad. But for a daughter of an affected male, they have, which is what we see here, they have a 100% chance of getting it. Again, we're going to talk more about this during Zoom, so don't panic. And then finally, for X-linked recessive, again, we're going to have XY. Oh, I like using the highlighter better for that. And XX are offspring outcomes. But now it's recessive. So for this pairing here, ooh, that's not the eraser. For this pairing here, that means this female must have been carrying it, because now her daughter has two affected X chromosomes. For her sons, here, they have a 100% chance of getting her recessive chromosome, because that's their only option. Now, if it was dominant, they'd have a 50% chance of getting that X chromosome from their mother. So for X-linked dominant, you'll often see more females than males. X-linked recessive, you'll see more males than females. This is a nice little cheat sheet that tells you, it's a checklist. It's just a checklist. When you are asked to figure out a mode of inheritance, you can go through and use this checklist to see where it would fall. Again, there's not that many Y chromosome ones, but you can use this checklist for sex-linked dominant. Here's some actual examples, like widow's peak, or if you can taste certain things. There are, as far as, now, again, most disorders, so this is something we're going to claim, or we're going to call diseased, or something that would cause you to have physiological issues. A lot of genetic disorders are recessive, strictly because if they were dominant, they'd get weeded out really fast. If there isn't medical intervention. With medical intervention, that might not be the case, because now we can see that there are dominantly inherited disorders. Achondroplasia dwarfism is such a disorder. Achondroplasia dwarfism is a type of dwarfism where you can't grow long bones. Your long bones are like your arms and your legs and your fingers and your toes. And these long bones can't grow properly, so you end up with disproportionate limbs to your torso. This is dominant, which means that even if someone that has the phenotype or is diagnosed with achondroplasia dwarfism has children with someone who does not have it, there is still a 50-50 chance that their offspring will have achondroplasia dwarfism. But on the flip side, there's a 0% chance that they'll have any carriers. What a carrier means is that you do not have the trait, but you are carrying the allele from the trait, and that is mainly for recessive disorders. Huntington's is another example of an autosomal dominant disorder. So if someone has a parent that later, and Huntington's doesn't really manifest until middle age, so that's one of the reasons why it persists in a population. And achondroplasia dwarfism doesn't affect your ability to reproduce. So for Huntington's, what you'd have is if one parent had Huntington's, even if the other one did not, there's still a 50-50 chance that they'd pass it on. Oftentimes, by the time it manifests, though, people are in their middle age and have already had children, and so that trait has been passed on. For recessive disorders, which what many genetic disorders are, you can have carriers, meaning that the turquoise here, it means is the affected allele, that some of the offspring are going to carry that trait but not be affected. Some offspring will be unaffected and not carriers, and some of them, if they receive both recessive alleles, will be affected. Sickle cell disorder is another example that has some really interesting evolutionary impacts. So sickle cell disorder, if you have a healthy red blood cell and you don't have sickle cell, it's just a nice round blood cell. A sickle cell will have a sickle shape to it, like a crescent moon shape. So in a homozygous individual, all homozygous recessive, all their hemoglobin is abnormal and they have sickle cell. There is something called heterozygous advantage, though, if you are in an area that has malaria. Because if you are homozygous dominant for sickle cell, you don't have sickle cell, but you are more likely to get malaria and die. If you are homozygous recessive, then you have malaria, and you will have physical weakness, pain, organ damage, even stroke and paralysis. So now you have sickle cell and are more likely to die. However, if you are heterozygous in an area that has malaria, your cells are just enough deformed where you still might have slight symptoms of sickle cell, but you're not going to die from it, and the malarial parasite can't live in your red blood cells, so you can't get malaria. This is then favored in the population. The good thing is that it helps prevent people from getting malaria. However, because it's a Mendelian trait, I'm going to say A is going to be the sickle cell trait. If you have two heterozygous individuals that have offspring, there's now a 25% chance their offspring will be homozygous recessive and have sickle cell. But there is an evolutionary benefit to being homozygous in that case, and that's called heterozygous advantage. Another autosomal recessive disorder is cystic fibrosis. It is a lethal genetic disorder, and it's in one out of every 2,500 people of European descent. And it results in quite a few symptoms, as you can see here. So it's polytropic. You get mucus buildup in the lungs and some internal organs. There's abnormal absorption of nutrients in the small intestines. It has quite a few traits that are associated with it, or symptoms that are associated with it. But again, it is just autosomal recessive, meaning you have to have that homozygous recessive individual to manifest it. So recessively inherited disorders show up only in individuals homozygous for that allele, and thus being homozygous recessive for that allele, like albinism, which is a lack of pigment. Carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal. This would be a carrier, this would be a carrier. Most people who have recessive disorders are born to parents who are carriers of that disorder. So their parents don't have it, but then that manifests. For consanguine, and consanguineous is the fancy term for inbreeding, because you have the same blood. If a recessive allele, there we go, causes a disease and it's rare, then chances of these two carriers meeting and mating is low. However, consanguineous matings increase the chances of mating between two carriers of the same recessive allele. I'm going to use Game of Thrones as the example. There's the Mad King thing. Apparently the Targaryens are like super into sibling marriage, and that's a recessive thing. According to this, Lyanna Stark is related to them. Never heard of that, but okay. And so this is a bad chart. Bad chart. To indicate consanguineous marriage, though, we use two lines. So if you have individual 1 and individual 2, and it's a consanguineous pairing, we'll draw 2 lines between them instead of 1. And you can see that here, and I'll show you a better chart in a second. Most societies and cultures have laws, or we have laws in America, or taboos against marriages between close relatives. It might be that, for the most part, people quickly realized that doing this results in trouble. However, at different periods in history, that has been more or less common too, unfortunately. So here's a good case. Here's a real case, not a Targaryen case, of consanguineous marriage or consanguineous mating. I don't want to say marriage. People mate without being married, and people get married without mating. So I don't like when people use them exchangeably, mating and marriage. When you mate, it doesn't mean you're married, and when you're married, it doesn't mean you're going to mate. But here's a case of consanguineous mating called the Blue Fugits of Kentucky. This is a real person. There's nothing photoshopped or anything here. You can Google it. So the Blue family of Kentucky. Martin Fugit and Elizabeth Smith were the original members of this family. They settled near, of course it had to be called Hazard, Kentucky, around 1800. Both were carriers of recessive metaglobin anemia, or MET-H, gene. So was a nearby clan. The Fugit's descendants intermarried. So you just had two people, or two families, that Hatfield and McCoyed themselves into the middle of rural Kentucky and then didn't leave. And what would happen is, if you have this recessive disorder, you have a hard time getting blood to certain parts of your tissues, and you end up with this blue coloration. They didn't know anything was wrong until some census workers came by. I don't know if anybody had a census. They didn't have a lot of people out this year because of the pandemic, but if you got your census, should have filled out your census. Some census workers came by, and they were, there to count how many people were there and how many toilets they had. And then they noticed, like half the people here are blue. So that's not good. And that's when it was discovered what had happened. So here's the pedigree for this Fugit family here. The blue individuals are going to be the ones that are carrying the recessive disorder that makes you blue. We have some carriers, and we have some probably heterozygous individuals. They weren't quite sure. So the reason that this incredibly rare disorder was so common in this family was that every single one of these circles is a consanguineous pairing, is a consanguineous mating. These are all married, breeding with and married to your relative, is what was going on with these red circles. And you can see how frequently that occurred in this one family. So multifactorial disorders is the last thing we're going to talk about. Many diseases like heart disease, diabetes, alcoholism, certain mental illness diagnoses, cancer, have both genetic and environmental factors. Lifestyle has a tremendous effect on the phenotype for cardiovascular health and other multifactorial characteristics. We call these multifactorial disorders. Genetic counseling is where basically people bring in their family histories, and then you can evaluate them for their likelihood to pass on that disorder. Here's a nice little cheat sheet. Another one. Looking at pedigrees. Now we're going to practice some pedigrees together. So let's start here with number 1. First of all, does it appear in every generation? Yes. Do affected individuals have affected parents? Yes. Are males and females roughly equally affected? Yes. So this is most likely autosomal dominant. And it is. This next one. Do affected individuals have affected parents? Always. No. Is it appearing in every generation? Yes. Are males and females roughly equally affected? So 1, 2, 3, 4, 5, 1, 2, 3, 4. Yeah, they are. So go ahead. Guess what you think it might be. And it was recessive. The reason is here. Once you have this, that an affected individual does not have affected parents, you know that it cannot be autosomal dominant, regardless of the fact that it's in every generation. It's in every generation, that's true, but this individual here does not have affected parents. Okay, next one. Do affected individuals have affected parents? Not always. Are males and females equally affected? Well, we have 3 females, but we have 1, 2, 3, 4, 5, 6 males. So I'm going to say no. Now, here's what you want to look at when you get to this instance. I have find an affected female if you can. Let's use a different marker color here. Okay, so I have an affected female right here. Do all of her sons have it? Yep. I have another affected female. Do all of her sons have it? Yep. Do her daughters have it? Well, it looks like, you can't see because I drew all over it. It looks like that her daughters don't. Now, this instance here is going to be your dead giveaway. Dad isn't affected, which makes sense, because obviously his son or his daughters don't have it. But mom is not affected, but her son is. But the only X chromosome, because I know from this ratio that it's an X-linked disorder, but the only X chromosome he got was from his mom. So it must be, if he got it, but his sisters did not, it must be X-linked recessive. So sex-linked or X-linked recessive. Sex-linked is the same thing because we realize that like Y chromosome disorders like don't really happen that often. Okay, final one. Is it in every generation? Yes. Do affected individuals have affected parents? Well, let's see. Well, here's an affected individual, affected parent. Affected individual, affected parent. Looks like, yeah. Are males and females equally affected? So I have 1, 2, 3, 4 males, and I have 1, 2, 3, 4, 5, 6, 7, 8 females. So we're back in that boat, right? So we have that ratio that seems a little off. So for my females, do all her sons have it? No, they don't. One does, though. Do all females, if a female has it, do all her, if a male has it, if a male has it, do all his daughters have it? Yep. Because a female will have 2 X chromosomes, a chromosomal female will at least. If one of them is affected, she could give either one to her sons. And we actually see that with this group here. This affected female gave her affected allele to this son, but not to this son, meaning that this is most likely a dominant sex-linked or X-linked disorder. This one could be autosomal dominant too. We're just assuming because of the ratio that it's probably sex-linked. Okay, so that will wrap it up. You do have a pedigree assignment, so go ahead and get started on that. I'll also give you some other places that you can practice these, and we'll go over it during the Zoom call, like I said. I would definitely recommend practicing these, however, because they will be on the exam. I won't ever give you one on a quiz or an exam like this one, where it could be one or the other. That's not fair. How would you know? If it could be either one, you have an argument for it. How would I grade that? I couldn't. But, well, I guess if I could be like, which both could it be? But that'd be mean. You'll get some of them more like these ones here, where you can clearly indicate which type of mode of inheritance it is. The last thing I want to point out is you might notice there's some Roman numerals. The Roman numerals indicate which generation you're talking about. So each generation is on a separate line. And these Roman numerals allow us to discuss which individual we're talking about. So you'll notice that the individuals are then numbered as well. So I have 1, 4 for this one. I'm talking about this individual. And if you can give me an argument on an exam, like, well, number, we're down here, let's say, and you could say, well, they're all affected individuals have affected parents. Or if, like, you know, you wanted to argue that this one is obviously recessive, you could say, okay, well, individual 2 is affected, but their parents are not. That's what that's for. Again, we're going to do more of these in the Zoom call, so get some practice in. We'll talk about it then. Until then, though, I will go ahead and go get started on your assignment. I will answer questions during the Zoom call if you have any, and I will talk to you guys soon. Bye.